Hi, my name is Monica Ponder. I'm an associate professor from Virginia Tech in Blacksburg, Virginia. And today I'm going to talk to you about our research about developing inoculation methods for Enterococcus faecium delivered onto whole peppercorns, black peppercorns, and also whole cumin seeds. And this is work that we began, that's funded by ILSI North America's Food Microbiology Committee. And we first began this work looking at developing a stable inoculation strategy for Salmonella. And last year we presented that work on Salmonella, and we were interested to see whether or not the same type of inoculation methods that were so effective for developing a very stable population of Salmonella on whole peppercorns and cumin seeds were also effective for having a stable inoculum for Enterococcus faecium. We began this study looking at 3 different inoculation preparations. The first were growing the cells in liquid in tryptic soy broth, and then incubating them at 37 degrees, then applying that inoculum directly to the whole peppercorns and to the cumin seeds, then evenly mixing that inoculum. All of those seeds are evenly mixed so that each of the seed has an even coating of the Enterococcus faecium per seed. The spices are then dried back down to their original water activity of 0.5 and held for 28 days. The second method of preparation that we examined were growing the cells on a agar plate surface of tryptic soy agar. And in those cases, they have large petri dishes. They were, again, grown for 24 hours at 37 degrees. And then the cells were scraped off the surface just using a spreader. And they were taken off, and then they were applied directly on to the whole peppercorns and the cumin seeds without any further washing steps within there. They were evenly mixed so that the cells would be evenly distributed across the surface of the peppercorns and the cumin seeds. And then they were dried back down to the same water activity. Our third method of inoculation was using the incorporation into a native microbiota biofilm that naturally forms around the whole peppercorn or the cumin seed when they are present in a growth medium suspension. This was performed for 48 hours at 37 degrees, after which the Enterococcus faecium was incorporated in this thick level at fairly high concentrations after that. For each of these 3, these are liquid type of inoculations where afterwards they did have to be dried back down to the water activity of 0.5. They were then held for a minimum of 28 days. The spices were then, the surviving Enterococcus faecium was then enumerated after 1, 7, 14, 21, and 28 days to determine the stability during that time period. And they were enumerated onto bile-esculin agar and tryptic soy agar. The reason that both media were used is that the native microbiota of both cumin seeds and peppercorn have a number of other lactic acid bacteria besides the Enterococcus that we had introduced on there. So if we did not use a highly selective media, like the bile-esculin agar, then we were not able to distinguish between our Enterococcus faecium from the other native microbiota on the seeds. Results of our study show that overall, all 3 of these methods are a good strategy for delivering the inoculum onto the inoculum of Enterococcus faecium onto both the spices. The broth inoculation strategy resulted in the fewest number of cells deposited onto there. And This was actually kind of problematic if you're looking for trying to achieve a 5-log reduction. This method needs to be further refined because not as many of those cells were deposited on there. It's much easier to achieve a much larger inoculum for larger inactivation studies using either the agar grin method or the biofilm incorporation method. During the shelf life experiment, overall the Enterococcus faecium was even more stable than the Salmonella across the 28-day study. There was a very small reduction, and similar to what we had seen with Salmonella, the inoculation method also influenced the stability and the recovery after 28 days. With the Enterococcus, we saw fairly small reductions. Our blue bars are the recovery of the Enterococcus from peppercorn after 28 days. Our orange bars are the recovery from cumin seeds. And you can see that when they were applied as broth, that the recovery was less. So it was less stable when a broth inoculum was used after 28 days. Overall, this tells us that the Enterococcus faecium behaves similarly to the Salmonella enterica tennessee that we are using to validate for peppercorn and cumin seed inoculation. So future work that's funded by ILSI North America will be doing vacuum-assisted steam inactivation of Salmonella and Enterococcus faecium.